Welcome to episode 70 of the Pulse of Physiology. I'm your host, Dr. Jim Davis, and today we're introducing acid-base balance, a critical physiological process that maintains blood pH within a narrow range. By the end of this episode, you'll understand the definition of acids, bases, and buffers, the importance of pH homeostasis, and the major buffer systems that help regulate pH. So let's get started. Acid-base balance refers to the regulation of hydrogen ion concentration to maintain a stable pH in the body. Normal pH for arterial blood is 7.35 to 7.45, with values below 7.35 indicating acidosis and values above 7.45 indicating alkalosis. The body must tightly regulate pH as deviations can disrupt enzyme function, oxygen delivery, and nervous system activity. Acids are molecules that donate hydrogen ions in a solution. Strong acids fully dissociate, for example, hydrochloric acid, while weak acids partially dissociate, such as carbonic acid. Bases are molecules that accept hydrogen ions in a solution. Strong bases fully dissociate, such as sodium hydroxide, whereas weak bases partially dissociate, such as bicarbonate. Buffers are molecules that minimize changes in pH by either accepting or donating hydrogen ions. The body has three primary buffer systems that help regulate pH. The bicarbonate buffer system, which is primarily an extracellular buffer, occurs when blood pH is low, such as in acidosis. This equation shifts right, increasing carbon dioxide exhalation and reducing hydrogen ion concentration. When blood pH is high, or an alkaline, the equation shifts left, generating more hydrogen to lower pH. This buffer system works closely with the respiratory system, which regulates carbon dioxide, and the urinary system, which regulates bicarbonate excretion and reabsorption. The phosphate buffer system is similar, and this helps regulate pH inside the cells and in the urine. And so this system will either pick up or release hydrogen ions to maintain pH stability. Then there are protein buffer systems that work both in the intracellular as well as in the blood. Proteins like hemoglobin and albumin function as buffers. Proteins can accept hydrogen ions when pH is low or release hydrogen ions when pH is high. Hemoglobin is an example of this in red blood cells, which helps buffer pH changes caused by carbon dioxide transport. So to summarize, normal arterial pH is 7.35 to 7.45, with deviations leading to acidosis when pH is below 7.35, or alkalosis when pH is above 7.45. Acids donate hydrogen ions, bases accept hydrogen ions, and buffers minimize pH changes. The bicarbonate buffer system regulates extracellular pH, while phosphate and protein buffers help maintain intracellular stability. Carbon dioxide and bicarbonate play key roles in acid-base regulation through respiratory and urinary compensation. So that's it for episode 70 of the Pulse of Physiology. I hope this episode has helped clarify the basics of acid-base balance and why pH regulation is essential for physiological stability. Come back next time for episode 71, where we'll explore acid-base compensation and how the respiratory and renal systems correct pH imbalances. Until then, keep questioning, keep exploring, and stay curious.